Okay, let's keep going. So before the break, we talk about the translation. How to use the codon to find the anticodon, and to bring the correct amino acid to finish the translation. So I want you to know the start codon, which is AUG, and which code methionine. So every protein starts from methionine, and three stop codon, UAA, UAG, and UGA. These three means finish the translation process. Okay, so translation, the first process is called initiation, and when the translation starts. So initiation start from the start codon, which is AUG. AUG. So ribosomes will find the AUG place and start. And the first tRNA gonna bind with the AUG has the anticodon for AUG. So they'll bring methionine here. And let's look at the ribosome. So how come it's so precise? Because on the ribosome, it only have two spots. So it starts from the AUG. So AUG sequence, okay, the tRNA bring the methionine here, and then the next three will be another codon. So another tRNA with the correct anticodon can only come here, and they will bind. So they will form the peptide bond from the amino acid. Then this ribosome move, and now the second one moved to here, and this one is empty. So every time you only have one empty space, and means only the correct amino acid, correct tRNA with the correct anticodon will come here and bind, and they'll form the peptide bond. So every time you only have two space, it's called the P site, the A site. So theoretically, when they form the protein, they're always the correct protein come and bind with it. So because of those codon, anticodon structure, so when the mRNA, they have the codon, and the tRNAs need to have the correct anticodon. That's the only one can bind with it. And then only the amino acid, correct one, will come to bind. Okay, so after initiation, the next one is elongation. So the ribosome will keep moving, start from the AUG, and it will just keep going down. It will just keep going down. And when keep going down, the correct amino acid, this space is empty, so the correct amino acid brought by the tRNA with the correct anticodon, and will come here, and they will form the peptide bond. So amino acid will keep forming a peptide bond. And this process is called elongation. So from the mRNA correctly pass through to the correct amino acid sequence. Because the codon anticodon structure, so only the correct tRNA, which bring the correct amino acid, can come to bind. And between the amino acid and amino acid, they form the peptide bond. So they will keep connecting. And the peptide bond get longer and longer. So that's called the elongation. So this process is called elongation. And until you reach the AUG, right? Oh, sorry, AUG start, the UAA, UAC, UGA, those three stop codon. And that's when the process needs to stop. So the mRNA totally has 64 different codon combination, because each nucleotide have A, U, G, or C. Totally you have 4. And you have 3, each 3 of them is a code. So 4 multiplies 4 multiplies 4 multiplies 3 times, totally 64. So the genetic code, there are 64 codon combination. And 3 codon is called a stop codon. Stop codon. I want you to know, because there are UAA, UAG, UGA. And there's only one start codon called AUG. And AUG code for methionine. So every protein actually starts from methionine. Okay, so there's the totally 64 different combination genetic code. And again, I told you I want you to know the start one, and there are 1 start and 3 stop. So these four I want to know. So that's the translation process. Because the codon in the mRNA and the anticodon in the tRNA, so there will be a perfect match. And every time, only the correct amino acid will come, and they will form the long amino acid sequence. And this process happens in the rough ER. So it's in a cytosol. And those amino acid sequence will stay in the rough ER. And technically, we don't call it protein yet because it needs to be transported to Golgi apparatus and folded to the correct structure. And then you have the correct protein. But that's the translation. So translation starts from the mRNA and eventually end with the 1D amino acid sequence. So start again, it starts from the start codon, that's the AUG. So that's when the ribosome come to bind with it. And each ribosome only have two sites. So the first site, the tRNA, has the anticodon. So the start codon is AUG, so their coding would be UAC. So the first tRNA anticodon is UAC. They're going to bring the amino acid, which is methionine. So all proteins start with methionine. And the second one depends on the second one. What's this code? What's this codon? And another tRNA with the correct anticodon will come. And now this, it will bring another amino acid. And now these two amino acids here, they will form the peptide bond. And they will connect together, form the peptide bond. And that's called the elongation process. So this process will keep going, elongation. And until it keep going down until it reach the stop codon. We have three, UAA, UAG, UGA. So once they meet the stop codon, it code for nothing. It code for nothing. So no amino acid will be brought by the tRNA to here. It code for nothing. So there is no more amino acid going to combine with it. And so this amino acid sequence will be released. And now we have a 1D amino acid sequence. So everything looks great. That's how the cells work to use the DNA to carry genetic information, produce mRNA, then produce protein. However, we know, okay, especially when you run this machine enough times, error happens. And there are different kind of error. So one kind of error is point mutation. That's in this DNA sequence. That's in the DNA sequence. Okay, one of them turned to be wrong. Say it's supposed to be a C and now you replace it with a T. Or it's a T and then now you replace with a C. And this is called a base substitution, also called a point mutation. And you find when they code to the mRNA, the mRNA will be wrong, right? And every three of this code is codon. So now the tRNA bring the with the anticodon here, bring the amino acid will be wrong. So now this amino acid will be the wrong one. And that's called a point mutation, or it's called a base substitution. And this kind of mutations happens. And it's not as serious as this one. This is called a base insertion. This is much more serious. Base insertion is now you add one more nucleic acid in. And once you have base insertion, you find after this, the sequence is totally wrong because you add one more. So now they're going to read this one, these three, these three, these three. And after that, the amino acid sequence will be totally wrong because the codon is wrong. So in general, the base insertion, and also you have the base deletion, you can delete one. Those is more serious mutation compared with base substitution. But the general idea is once you mess up the DNA sequence, the RNA will be wrong, mRNA. And once the mRNA is wrong, the protein will be wrong. So the point mutation, one textbook example is the sickle cell anemia. Sickle cell anemia is an example of point mutation, means it's the base substitution. You should take T and replace with A. So what happens is when this happens in the DNA sequence, only have one substitute being replaced. And then the mRNA sequence is wrong because it's supposed to be a GAG and now it becomes a GUG. And when this happens, the tRNA recognize this codon. These two are different codon. So the tRNAs recognize this one and the tRNA recognize this one will be different tRNA. They carry different amino acid. So now you found the amino acid sequence. The amino acid sequence is different. This is called a point mutation. Only have one substitute replaced. And then those amino acid sequence only have one amino acid is wrong. But amino acids, some of them are hydrophilic, others are hydrophobic. So when they fold it to the correct 3D structure, no, it's not correct anymore because they will affect this 3D structure. So it turned out those protein have not correct 3D structure. It won't be able to perform its function normally. This is sickle cell. And this kind of red blood cell supposed to look like a donut. So in the body, we have the capillary. They are very tiny. They are like the inner diameter of the red blood cell. And this kind of donut-shaped red blood cell, smooth, they can go through those tiny capillary, carry oxygen to every living cell in your body. But not this sickled red blood cell. They could not. So their oxygen carry ability being compromised, and that's anemia. So it's sickle cell anemia. But they can still carry oxygen. It's like, okay, you supposed to have those good truck to carry those oxygen. Now you have those broken truck. They can still carry oxygen. But when the body need more oxygen, say when they start to exercise, they need more oxygen. And this kind of sickled red blood cell, they could not carry enough oxygen to the body. That's anemia. That's anemia. So the people with sickle cell anemia, they can still survive. It's like anemia. They just don't have enough oxygen when their body need more, desperately need more. So they can still survive. It means they can still pass their gene to next generation. So we still have the sickle cell anemia case in human because of this. So they show you this transcription translation process need to be perfect. If not, you can have mutation. And when the mutation happens, because the sequence goes from DNA to mRNA to protein, so if the DNA is wrong, we call them base substitution and also called the point mutation. What happens is the protein sequence will be wrong and you don't have the correct protein. And some mutation is the mutation. Some mutation have those insertion, you insert one, or deletion, and which cause much more serious mutation because the whole sequence after that will be wrong. And in some serious case, and the embryo won't develop. Okay, let's stop here.